Okay, so our final speaker before the break today will be Upi Bhalla, and he's representing a third continent in this very geographically diverse session. He's from Bangalore, India. And I remember actually first hearing Upi give a talk when I was a new faculty member at Cold Spring Harbor, and I was really amazed by his description of olfactory behavior, and it really awakened me to what an interesting and really important sense that is, and also how it's quite different from other sensory modalities that I had studied in my own training. So thank you very much for being here.
Thank you, Anne, and thank you, Tony, and everyone who invited me to this completely amazing conference. I've had great fun and I've learned a huge amount. I want to thank also all the people who've been running around making things work. And I want to make a special thanks to all of you in the audience who I have to say have been quite heroic, coming in at 8 a.m. and sticking around till 9 p.m. on the weekend. Well, great for you. Okay, so this is where I work, the National Center for Biological Sciences, which is a daughter institute of TIFR, and we are located in Bangalore. And we study biology across all scales, right from molecules to ecosystems. And I'm interested in studying, of course, the brain. Just before I launch in, here's a plug for a summer school that we are running again for the 5th iteration now. It's the Computational Approaches to Memory and Plasticity camp at Bangalore. And this year we have a special focus on sequences, which will also be the focus of this talk. Okay, so this is a classic diagram. Terry Serjanowski bears some of the blame for this. So of course, we all understand that computation in the brain happens across many, many levels. And you've heard a lot of stuff at these levels, and I'm glad to say that there has been some indications of interest down here. I'm going to take us right down to the bottom to the level of molecular computations with some hints of dendritic scale computations. Okay, so I'll start out by making a plug for molecular computations as being something which are extremely powerful and completely ubiquitous. And as you'll see, I will argue that they may play a bigger role in brain function than some of the other stuff you've been hearing about. Okay, so here's just a sort of very brief summary of some of the computations that happen absolutely routinely in your brain all the time. Neurotransmission, even though we think of it as an electrical thing, actually the really interesting stuff is happening at the synapse, which is some absolutely crazy cell biology and chemical signaling. And that happens very, very fast. It has to happen faster than, in fact, the overall time scale of information transmission, because this is just a small part of the cascade. There's neuromodulation, which you've heard about, various forms of adaptation and homeostasis in the levels of transmission, and this kind of leads into plasticity. You've heard quite a bit about that during this conference, ranging from short and long-term storage to changes in excitability, restructuring of connections, even rewiring of connections. And then we can think about kinds of molecular computations that happen in decision-making, ranging from pattern selectivity that are involved in different kinds of plasticity, to spatial patterning, like synaptic tagging, and sequence discrimination, which is what I will start to come to. But before that, let me make a little propaganda plug here, which is say to the effect that molecules actually do most of the computation in your brain. And it goes like this. Acknowledge that electrical computation is pretty fast, of the order of 1 millisecond. And here I'm leaving out synaptic transmission, even though I assert that that is obviously happening at least as fast as that. The thing with electrical computation is that it spreads over a fairly long length of a dendrite. The electrotonic length is lambda, the length constant is roughly of the order of 0.5 millimeter in many cells. And so that gives you some 5 or 10 zones which will have different electrical potentials across a cell. Compare that to chemical signaling, which is slower by and large, but because it's now limited by the diffusion constant of molecules, the number of zones is very much smaller, and on top of that you have this beautiful compartmentalization that takes place in all synaptic spines. And so you have some very, very small scale, short length scale computations that can happen. And these two terms, as you can see, cancel each other out nicely. So at that level, You can do about as much number crunching in the chemical as in the electrical domain. But the key thing here is that there are hundreds, if not thousands, of different signaling molecules and pathways active at any given time, whereas for electricity, it's either potential high or low. So this gives us a scale advantage, which I assert means that molecular computation is very, very powerful. Okay, so that's just a plug for it. We are all familiar with different levels of chemical computation in the body as a whole. There's the DNA, RNA, protein sequence, which we're all familiar with. And computation happens at all of these levels, gene interaction circuits, more recently RNAi-based interaction circuits, and then the somewhat more familiar biochemical and protein signaling cascades. I'll be talking mostly about this, but in reality, these are all across. They interface with each other all the time. It's quite seamless. There's no boundaries between these. So this is a set of pathways I've been working on for very many years. In fact, I did my postdoc at the other end of town with Ravi Iyengar, looking at this set of pathways involved in plasticity. And if you take the trouble to figure out what kinds of reactions comprise each of these little colorful boxes, you'll discover that there's a fair number of reactions in each of those. And this starts to get somewhat messy when you want to actually graphically analyze this and simulate it. So this is a complexity not so much of the millions and millions of computations that one does in deep learning. It's a complexity in terms of there's a huge diversity of different rate constants and different molecular properties, and yet underneath it, it's the same equations. It's chemistry. It's reaction to fusion chemistry with perhaps a little bit of stochasticity thrown in. So it's complicated because it's diverse, but fundamentally the principles are still pretty much the same. So here are some of the things that you can do with molecular computation just at the synapse, and we mentioned some of these: transmission, associativity, memory storage. I'll come back to that in a moment. Molecular traffic, pattern selectivity, dendritic excitability, synthesis, and even restructuring this. Let me spend just a few minutes talking about memory storage as sort of my entry point into looking at molecular computation. So this is a very famous painting. Anybody remember what its name is? It's Persistence of Memory. It's Dali's Persistence of Memory. And the reason why I like it is that actually, if you think about what's going on in a synapse, in a dendritic spine, this is sort of the epitome of impersistence of memory. It's a very bad place to store information. And it's bad for multiple reasons. One is that the volumes here are so small that the number of molecules that participate in any reaction, including the reactions that store information, are minuscule, of the order of a few digits, tens to hundreds at most. And with so few molecules, you're going to have huge amounts of chemical noise. And this will simply erase your stored signal if it doesn't erase it even as it comes in. So stochasticity messes up the computation at the synapse. Turnover is another problem. It's a bit of an alarming thought that the very molecules that make up your brain aren't going to stay there for more than a few days at most. So if that is the case, how do you remember anything past the few days where your last few molecules trickle out? Okay, so fortunately you do. There are ways around this, but we'll see that in a moment. And the other is that if you just put a molecule there and hope that it'll stick around, it won't. It'll simply diffuse away in a matter of seconds. And so now you have to also interface the chemistry with some interesting structural changes in order so that the molecules stay where they're put. So all of these mean that it is actually a very bad idea, at least at face value, to store information in something as fragile as a synapse. And yet, that's what we seem to do. So what's happening? So here's a proposed solution to the problem of turnover, of the fact that your molecules simply do not last very long. So let's suppose that we have a nice little feedback loop. A activates B, B activates A. So let's say the blue color means that they're at a resting stage. And you give it some kind of a kick, a strong stimulus, something you want to remember. And now they're in a high activity state. And you can think of that happening at the level of the individual molecules in the system. Okay, very well. So now these gray blobs are meant to represent synaptic spines. Let's suppose we have stored some information in this spine here. And of course, through the ravages of time, a few molecules get degraded. And if it were just to continue going downhill, you wouldn't have very much left. But of course, the cell is alive. It's a non-equilibrium thing. There's a lot of stuff going on, and so fresh molecules come in. But these fresh molecules are ignorant. They don't know anything about what was going on here. But what happens is they come in, and it's just a small perturbation in a state of excitability. And so these get recruited to doing whatever the other molecules there were doing. And so you retain the information even though you may lose the molecules. To analyze this a little bit more precisely, if you have a feedback system, and supposing you only consider the interaction from B to A, you only consider this part of the feedback cycle, you can construct something which is very familiar to and favored by chemists, which is a dose-response curve, right? You set a certain level of B and you estimate the resultant level of A, and you get a curve like that. And you can do it the other way around. You can change the level of A and get a certain level of B. And as all of you mathematically inclined people know, if you want to now get the stable points of such a system, you take this and you flip the axes so that they sit on top of each other, sort of rotate it around the 45-degree line, and you get intersection points, which can be a single point like that. So that is a stable point of the system. And we can discuss offline how you might end up with this being a stable point. Basically, you start anywhere here, you will sort of bounce back and forth between these lines, and you'll always end up there. The interesting thing happens if you have multiple intersection points, in which case there will be a lower stable point, an upper stable point, and an intermediate transition point. And this is just a bistable system, which, as most of us know and love, is the staple for information storage in your computers. So you can get a bistable system out of chemistry. And this is what I'm arguing is what is happening in such a system as this to allow you to store information for long times. And this is a system which I looked at, which I studied as a postdoc and after that. And you can get it in low state, you can flip it into a high state. You can even sort of balance it right at the edge in the middle in this intermediate state, in that transition point, and it can, with a small transition, it can go one way or another. So this is a bistable system. I haven't yet addressed, and I won't, in this talk, I won't talk about what happens with the chemical noise. That's actually a very, very interesting problem, and it's been analyzed by many groups, including our own. But suffice it to say that if you take the same switch and throw in chemical noise, you get the very unfortunate situation of state flips spontaneously, which is not a good thing. There are ways around this, but that's a story for another day. Okay, so these are some of the molecular computations, and let me now then take you to the core of what I'd like to talk about, which is sequence discrimination. And this is really interesting. It's something which I'm very glad many speakers have brought up. David Tank brought up, several people brought up over the course of this symposium. And people have, what we've heard about mostly has been in the domain of generation of sequences. What I'm going to talk about is the recognition, the detection and discrimination of sequences. And that is where molecules sort of shine, because they're actually able to use time and delays as a form of computation rather than it being an impediment to computation. Okay, so let me start with sequence computation. So this is, as you've already heard, there are many kinds of sequence activity. And just to recap, if a rat, for example, is running across a linear track, there'll be place cells which will be active at different locations along the track. And because the rat's movement is in a sequence, you get a sequence of activity. And you also heard from Ela and others that you can get replay, forward and reverse. That is, this whole sequence can happen again in a shorter time scale. We've also looked at time cells. This is from Modi and from Ashish and Mehrab in my lab. They looked at a different kind of sequence formation that occurs when rats, I should say mice, do association of two stimuli, and a sound and an air puff. After learning, there is a sequence of activity that forms. So sequences are very ubiquitous, and there have been various analyses on how one can process these. Dean Buonomano has looked at, and various other people, in fact, there were some mentions of it in the symposium, have looked at ways in which networks can perceive, detect, and recognize sequences. And Mike Hauser, who I'll talk about in a moment, has come up with a nice electrical way in which this can happen. What Mike Hauser did was, Tiago Branco in his lab did this marvelous experiment where here's a neuron, and what they did was they homed in on a small dendrite, a small branch of this, zoomed in over here, and then they used glutamate uncaging to stimulate at successive times different locations along this dendrite. So 1, 2, 3, 4, 5, 6, 7, 8. So in a very rapid succession, they illuminated these points. This caused local release of glutamate, which caused local activation of the dendrite. Okay, and what did they find? What they found was that even this small subcellular region was sequence selective. That is, that you got a strong response if the stimulus was going from outside in. In other words, if it was starting far away from the cell body and moving towards the cell body, there was a larger response than if the stimulus was starting the cell body and going the other way. So here it is larger, the red curve, and going the other way, it is a much smaller response. And so the cell, in a subcellular way, can discriminate between two sequences, going one way or another. Now, this is actually a very rapid process. This happens on the time scale of roughly 40, 50 milliseconds. And we were very interested in asking, is there a way in which one can perform a similar kind of computation, but at behavioral timescales? And so we turned to molecules. And this is the general framework of what we're thinking of. It's actually a very straightforward idea, and based, as you will gather, on lots of existing work. And the idea is simply this. If you trigger events, let's say through some projections to this schematized dendrite, so here's a synaptic input to this spine, to the next, and these fire in order. If you do this in such a way that there's some chemical signal triggered at each location, and this chemical signal propagates. If it propagates, and if successive inputs come just at the time when the propagating wave reaches the next location, it's like you have a traveling wave and you're boosting it each time it goes along. So if your speed of propagation of the chemical wave itself is matched by the timing of the arrival of inputs, then you can boost this up and get a strong response. Whereas if you do this in some kind of scrambled, disordered way, the little kicks do not contribute to each other, and you don't get a strong response. Okay, so to analyze this, we made a bunch of spherical cow models, which are simply models which have the absolute minimum, ridiculously minimized level of a number of parameters. In this case, each of these had only 2 state variables. And some of these will be familiar to you. I'm not asking you to recall the equations. For now, we'll just focus on this, though this one, which is probably also familiar to you, is the Fitsu-Nagumo form. And this also does the same thing. So this then is the configuration. So we have a stimulus that sweeps along at a certain speed. And every time it does so, here it goes again. So it sweeps along at a certain speed, and it therefore triggers the activity of the readout molecule of the chemical cascade somewhere else in the same dendrite, somewhat lower down in the chemical sequence. Okay, so what happens here? So the basic outcome is that this very, very simple abstract system is capable of discriminating sequences. If you give it the sequence in the correct order, you get a nice strong buildup. If you give it in some scrambled order, the buildup is much smaller. Let me just show you an example of that again. The blue curve is coming in sequence, as you will see that the little blips are coming in order. And you can see that it is much, much larger than the red curve, which is out of sequence. It is disordered. I'll just let it run one more time so that you can see what's happening. So blue is in order, red is scrambled. Okay, so what you see then is that this very minimal chemical system is capable of discriminating ordered from disordered sequences. And in fact, since there are 5 inputs in this example, there's a huge number of disordered sequences, and only one, actually turns out there are 2 possible sequences which are ordered because it's completely symmetric. You can go either way. So it's very good at discriminating order from disorder. So that's nice, but this is a very reduced system. So I really like this slide. This is sort of a shock and awe slide. This is half a micron across. This is a marvelous image from Sven Truckenbrot. And what he's done is he has illustrated the packing of proteins inside this very, very small structure. And the point is that every single one of these proteins is potentially doing computation for you, right? So it'd be amazing if we were able to simulate this. We can't, but it's just a reminder of how much computational machinery there is, even in one, this is a presynaptic bouton, even in one presynaptic bouton out of the hundreds of thousands that you have per cell, tens of thousands you have per cell. Okay, so we didn't go quite so extreme, but what we did do was to put a set of reactions, some of them belonging in the postsynaptic density, some of them belonging in the bulk spine, and some of them belonging in the dendrite, and we scattered this across a cell. And this is a morphologically detailed cell. In fact, we tried multiple cells. It doesn't really matter very much. We had some 5,000 spines over this. Every single one of them had all these reactions. They were all coupled in this lovely way. All of the electrical stuff was also happening in this. There was background activity. I mean, basically, we threw the kitchen sink at this. Here's a picture of what the outcome looks like. I'll ask you to look for the little yellow spots, which are simulated calcium influx at each of the synaptic points. So this is just random background activity. This is what all of your neurons endure all of the time. And this is not even very fast random activity. It's 0.1 hertz average activity across the cell. So the challenge I want you to sort of pose to yourselves is to ask, if there is a sequence of activity in all of this, could you detect it? Yeah? You with your marvelous visual system, which can do all this massively parallel processing, can you spot the ordered activity anywhere in this mess of random activity? And the answer is it's extremely hard to. But the point is that this is precisely what having all of this chemical activity in the cell is good for. And so what I'll show you in a moment is just that we have very small segments of dendrite onto which I have explicitly delivered ordered sequential input. And what you'll see in the next slide is a comparison of the case where there is ordered versus disordered input. Okay, so we're going to zoom in. Just to remind you, there's the whole complicated cell, and this is the small region over which the sequential input and the non-sequential input is being given. And it'll just run by again. And what you can see is that the sequential input builds up nicely. The disordered input fades away. The point being that even when we throw in all of this detail, even when we have all of that background activity hitting the cell, the system is still very able to select and respond to ordered as opposed to disordered input. So it's a very good sequence discriminator, and it's doing this in parallel over this entire complicated dendritic tree. Okay, so let me now turn quickly to some very recent unpublished work where we've analyzed how likely is it that cells will do this in a network. So the question is, supposing you have an ensemble of neurons which are connected in such a way to a given dendrite. How likely is it that you will get this precise order of connections, both spatially and in time, to a given cell? And at face value, this might seem extremely improbable. So we crunched the calculations, we did some simulations, we did some math. It turns out that if you do not have a requirement for sequence ordering, the numbers aren't so bad. You end up with fairly high, something of the order of 10% of cells will have clusters of synapses which are composed of these inputs. That's not very helpful. You have far smaller, but still not completely unreasonable in a large network, you have far smaller numbers of cells which will have the correct connectivity. The different colored lines are for different kinds of network statistics that we analyzed. And these are certainly in the range in which you could get something useful. However, that's not the only thing going on when you have sequential input. For example, this is an analysis that turned out, well, it turned out okay in the end, but it was a little bit alarming, which is that if you get any inputs purely at random that interrupt the sequence, it completely screws up the response. Not surprisingly, right? You have a system which is tuned very specifically to respond to a given sequence, and now some random activity comes in, random synaptic activity comes in, it's going to get confused. And that's what this analysis says. So we have our regular input coming in, and we tried different points at which we would deliver a random input at the wrong time. And it turns out that it doesn't care very much if the random input is outside the zone of sequential input, but once it's in that zone, the selectivity just plummets. It becomes a very bad sequence discriminator. So this is a problem because, of course, there is random background activity in all neurons. How do we avoid this? Well, the short answer is we threw this into the calculations, we turned the handle, we did a whole lot of other calculations, also thinking about cellular geometry, connectivity patterns in the network, background activity, connection stats, false positives. Sometimes you can get the cell reporting a sequence when there wasn't any. So all of these things put together. And what we found was something kind of cool, which is that there's a sweet spot for different kinds of networks. That is, some networks like sequences, I'm anthropomorphizing here, but they like sequences of length of the order of 2 or 3. Some like longer sequences, and this seems to work pretty well for different kinds of network statistics. So here's a more pictorial view of exactly the same calculation. In the hippocampal, using Mike Hauser type sequence selectivity, it likes shorter sequences. The little black dots are sequences of length 2. The little blue ones are sequences of length 3. In the hippocampus, using calcium-induced calcium release sequence selectivity, very, very sparse recognition of any sequences at all. In the cortex, there's different patterns of activity. It likes somewhat longer sequences in the electrical domain and quite long sequences in the chemical domain. So this is all actually quite remarkable, I thought, that you could get, on the basis of network properties, statistics, you can get different kinds of sequence preferences. So the point of this is that here you have a fabric that is purely at random. We've not done anything directed about the connectivity here, but you have a network in which a large fraction of cells is selective for certain sequences. And over the population as a whole, you will have some cells that like that, some like something else. So given a reasonable range of ensembles of different kinds of inputs, you will very likely have cells that respond to pretty much any permutations of those inputs. And these are just the responses of this network to one given sequence, but you could imagine different populations, subpopulations of cells responding to other kinds of sequences. So it's like the network is ready there to pick up sequences of pretty much any order. Okay, so let me now end with a little bit of speculation about how this might map to some of the deep learning and AI kind of ideas. This will be familiar to you as a representation of the LSTM. So you're all familiar with the wonderful and terrible things that these networks like to do to you. They like to talk to you, translate for you, and sell you things. Yeah? Okay, so wouldn't you like to make them even better at it? Yeah. So well, that's what this meeting has been about, right? So now what is the trick? How do you get time into such networks? Well, the trick has been there for a long time, which is that you start with your classical feed-forward network and you introduce a feedback step, and that allows you to introduce history because the past state of the network is now part of the future computations. And this is usually unrolled for reasons of being able to do this massively parallel. So you just take this feedback thing and sort of make duplicate copies of it that happen at different times. And this is all, sorry, I'm just rehashing familiar stuff to most of you. Okay, so this is the unrolled network. And you can replace these things, klutzy things, with a much more flashy LSTMs, which do all sorts of clever ways of handling. So these are important because they are very good at storing past state of the network in a manner which facilitates its computation. And one can make speculations that, okay, you know, this looks a little bit like the hippocampal entorhinal cortex loop. If you wave your hands sufficiently vigorously, you can convince yourself of this. Yeah, fine. But I'm going to go much deeper than this. What I'm going to suggest is that let's look at what a single neuron can do. And of course, a single neuron is out there. It's receiving inputs from all its dendrites, yeah, all its 5 to 10 millimeters of receptive area. And each sequence only needs to be 20, 30 microns long. So you have a lot of possible sequences it can recognize. So taking a cue from Bartlett Mell's work from many years ago, every single junction, every single part of the cell could potentially be doing a kind of summation and a threshold function of its inputs. But it can be doing more. You can have gating, you can have inhibitory signals shutting off different parts of the dendritic tree. That's cool. And what I'm arguing from this study is that you could also have each of these little pieces of dendrites doing a lot of sequence recognition all in parallel, right? And of course, you have, these are the same molecules that are, of course, involved in learning and memory, in plasticity. And so you can have weight changes occurring all over the place. So now this starts to look like a pretty formidable computational machine doing time domain computations and sequence recognition. So I'm saying maybe there's some level of similarity or parallels in the kinds of computations you can do with this one neuron compared to an LSTM. Of course, this is all happening within the cell. It's all local, but there's only one output. So that is not quite the same thing, but I just throw this open as a suggestion for those who like to think about such possibilities. Okay, so to wrap up, we've looked at molecular computation, we've looked at sequence computation at different scales, and I've tried to make the case that this is actually absolutely ubiquitous. A completely random network will exhibit this. And possibly there are parallels with what even single neurons can do and what nodes in the deep learning network might be able to do. We've seen sequences happening at all sorts of scales: behavioral, systems, cellular, and subcellular. And what I'm going to suggest is that sequence recognition computation is absolutely pervasive, that the statistics of natural scenes could be encoded because you have the possibility of having sequence recognition exhibited throughout the network in different combinations, and that this might have some kind of interesting mapping to the roles that they play in brain networks. And molecular computation, subcellular computation underlies both of these. Okay, so I'd like to thank all of you, and I'd like to thank the gang, especially those people, and the people who have funded me.
Great, thank you very much for that beautiful talk, and we have time for some questions. In the back?
So in the beginning, you showed like very complex network of like molecular interactions inside the cell.
Mm-hmm.
And do you think those networks and those like algorithms are more like kind of hardwired, largely hardwired like circuits in, for example, like brainstem, or it has some learning capabilities? Is any evidence showing that, like generic learning capabilities?
So that's a very good point to bring up. They are hardwired in the sense that they are genetically encoded, and each neuronal type will be expressing some fairly large subset of those. However, they are also extremely dynamic in that different regions of the cell might have, through their history, expressed some subsets, some proteins, some receptors at a greater level than others, or there may be different phosphorylation states in different locations. And in fact, these molecules are in fact the substrate for the synaptic plasticity that underlies a lot of the computation that you've heard about throughout the symposium. So it's hardwired in the sense that the topology is reasonably well defined, but it is very, very flexible in terms of the expression and phosphorylation state.
Terry?
So your chemical dendritic computation reminds me of synaptic tagging and capture, which you also mentioned early on.
Yes.
Which is very fascinating because of the time scale involved, right? So the idea is that if you have a synapse that is weakly potentiated, so it will eventually fade away in terms of its increase, next to a strongly potentiated synapse, that the weak one will be rescued and will become strong. What's amazing is that the time window is an hour, plus or minus an hour. So if this is what your chemical system is doing, being able to, in a sense, what's the right word for being able to? By the way, the spatial scale is more like 10 to 20 microns, so it's not the whole dendrite. but it could produce cohorts of synapses
Yep.
that have somehow are encoding similar things. I'm not sure about the order, but now you could imagine having your order sequence extended further along the dendroid.
Absolutely, Terry. Yeah, thank you very much. That is one of the ideas that we think is an interesting route to go down when thinking about this. That is that these are in fact the molecules that are involved in plasticity, and there's already evidence from a lot of this wonderful synaptic tagging work that plasticity spreads out in local domains. So we think this is absolutely where things are likely to happen. Yeah.
So some people have proposed.
Sorry, where are you? Yes.
Hi. Some people have proposed that there can be both positive and negative interactions. Terry's focusing on a positive interaction that takes a long time. There's been proposals of negative interactions between synapses where a strong synapse can steal from its neighbors.
Mm-hmm.
That's another way that this can happen, and we've seen cell biological evidence for this happening on a timescale of minutes,
Mm-hmm.
involving stealing of glutamate receptors from
Mhm.
neighbors,
Mm-hmm.
and so that might be something useful. One thing I liked about your talk is that you were frank about admitting difficulties, and ectopic inputs was one thing that you pointed out could kill the Bronco-Heuser phenomena.
Mm-hmm.
What if it turned out that there was a socialness to the way the inputs came to a local stretch of spine, so that the ectopic synapses were systematically expunged because of some kind of learning rule? This is a way that, you did some calculations based on random connections, but what if the plasticity was actually encouraging the ensemble behavior? Then the whole rules of the game would be changed. Have you modeled that?
Yes, we modeled that. So that's a great question. So it is a real concern that you would get ectopic input and then lose the ability to select for any sequence. So the thing is that random ectopic input, it's very hard to come up with a learning rule that will allow you to selectively nullify that, especially given that there's so many potential overlapping sequences. However, a very important category of ectopic input is that the same ensembles are sending additional synapses into this selective zone, but they're sending these axonal projections to the wrong place. Those are the connections that we've analyzed, and those can be eliminated through appropriate learning rules. In fact, very much like SCDP type learning rules. So we were not able to come up with a way to eliminate completely random background, uncorrelated background activity, but we can eliminate correlated activity and eliminate that as a factor that spoils the selectivity.
So we know there are fast sequence during shortwave variable and the behavioral timescale sequence. So how does your chemical computation sequence recognition model can recognize both of these sequences?
Yeah, okay, so you really pinned me down. Okay, so that's a very, very important question. So yes, there's a whole range of time scales that one needs to worry about. And I only mentioned the time scale of Mike Hauser's roughly 50 millisecond sequences. It turns out that one can come up with almost the same kind of propagating mechanism using calcium-induced calcium release, which is relatively fast. That's of the order of half a second or a few hundred milliseconds. And one can also come up with a similar, at least logically similar kind of computation using more conventional kinase cascades. And that is slower. That is on the 10 to 15 second time scale. One could go even slower in principle and talk about protein synthesis cascades, but I would much rather try and establish experimentally some of these faster ones before extending the speculation that far. But it's a great question. Yes, one does have to worry about multiple time scales.
Very, very nice question, but I was wondering, for these very slower time scales that you mentioned, then you give more time for some these noisy inputs to come into to disrupt it, right? So
Yep.
it is might be good for the short time scales, right? Because there's not so much time for noise to kick in, but if you're going to wait like tens of seconds, then other things might kick in. And
Okay, those were part of our calculation. So it turns out for the electrical sequences, the time scales are shorter, but the length scales are longer. So there's more locations at which you can get ectopic input. And furthermore, for the electrical sequences, you can interrupt the sequence by ectopic input even outside, but within the length constant, within the electrical length constant range of the actual sequence delivery. So it's a bit of a push and a pull. So even though one may be fast, it doesn't necessarily escape the problems of ectopic input, and the other one is sort of rescued by it being very, very slow. So by very, very short. So there are trade-offs, and these are different trade-offs for these different cases, and so that's why you end up with different networks have different preferences for the kinds of sequence lengths that they like. Where did that slide go? There we go. Yeah. So you have different kinds of preferences depending on the statistics of the system.
Okay, great. Well, let's thank our speaker again.
Thank you.
Great talk.